FAMU's Center for Viticulture and Small Fruit Research is the premier research center studying American native grapes in the southern United States, and there are many exciting opportunities for students to partake in scientific research here. The breeding program, led by Dr. Islam El-Shakawi, is aiming to develop and release new grape cultivars with superior qualities as fresh fruit or sources for new styles of wine. Here, students are engaged in studies of fruits from different varieties of grapes grown in our research vineyard. We utilize various instruments to evaluate the fruits, such as these calipers, fruit hardness tester, and colorimeter, which measures size, firmness, and color of each berry. Juice characteristics are also important for evaluation, especially for potential wine grapes. Even the seeds are removed and evaluated. The flesh of the berries is then frozen with liquid nitrogen and stored in freezers to preserve them for future research studies. In the Viticulture and Product Development Laboratory, led by Dr. Violetta Silova and Mr. Frank Humphreys, we study how to better grow grapes in Florida and what value-added products can be made by the bioprocessing and vinification of grapes. Our research program includes the Wine Analytical Laboratory, where experimental wines made here at the center and others submitted from wineries across the southern U.S. are analyzed for various important vinification characteristics such as pH, acidity, sugar, alcohol content, and overall healthy qualities. Student researchers gain valuable knowledge and experience with chemistry, food science, and principles of fermentation. This analysis being performed will tell us which grapes and wines may have healthy qualities associated with them. Antioxidants found in foods such as the grapes can protect our bodies from oxidation and prevent cancer and inflammation. This research is being used to assemble the digital reference database for Southern wines, which can be utilized by commercial wineries and researchers as a standard for quality comparison. Our genetics program is the only program in the world solely dedicated to studying the genomics of American native grapes. The molecule of life, deoxyribonucleic acid, also known as DNA, is extracted from the berries and other parts of the vine, such as these leaves. Liquid nitrogen is used to freeze and dry the sample, which facilitates grinding with the mortar and pestle. After sample preparation is complete, genetic material can be extracted by following a specific protocol. When extraction is complete, the samples can be stored for future analysis. Molecular techniques including gel electrophoresis and gene sequencing can be used to determine which vines possess the genes responsible for beneficial characteristics such as drought and salt tolerance or capacity to tolerate pathogens. The potential new varieties for fresh fruit and wine production can be released in the future by the university to be utilized by the industry. Students have a great opportunity to learn cutting-edge research technologies while gaining valuable experience and training which prepares them for future academic or science careers. In Vitro Culture Lab is another exciting student training area where young researchers can experiment and learn. We grow and maintain grape cell cultures inside these petri dishes and test tubes. These plants and plant cells grow under sterile conditions which keeps them safe from diseases which affect the plants outside in the environment. The purpose of cultivating these cells is to produce material for further analysis and experimentation at the single cell level. The small plants are grown in order to produce disease-free planting stocks, which will eventually mature and be moved to the greenhouse and the vineyard. Plant Biotechnology Lab is led by Dr. Sheikh and focuses on improving berry characteristics and product quality by using biotechnology techniques and equipment to analyze protein and DNA. The red media you see here contains extracts from the muscadine grapes grown at the center. The extracts contain phytochemicals which exhibit anti-cancer properties. Cancer cells are grown on this media in petri dishes to determine the effectiveness of these phytochemicals at fighting cancer. Students interested in cancer research are able to study these phytochemicals. These images are called two-dimensional proteomic gels, which let researchers visualize what proteins are present. Different proteins produced by the grapevines can protect them from diseases or make them more susceptible. This research enables the production of new value-added products that can be utilized by growers and the industry while also enhancing consumer health. The center's 45-acre research vineyard is home to thousands of different experimental varieties of grapes. Under the dense canopy of leaves, you can find the berries that all our research is focused around. The grapes come in all shapes, sizes, colors, and tastes. Some grapes that are fit for winemaking are processed into experimental wines using various equipment such as this wine press, which squeezes out the juice and leaves the skins behind. Afterward, yeast is added, which begins the fermentation process. We hope you enjoyed this video and are interested in learning more about the vineyards and the research that goes on here at the Center for Viticulture and Small Fruit Research. We look forward to meeting you and working together in the future.